Hello, everyone. I'd like to thank the organizers for giving us the opportunity to present our work today. So as everyone knows, PAX6 related aniridia is a haploinsufficiency, meaning that one copy of the gene is not working. So the effective dose of the PAX6 gene is reduced, which then has consequences during the development of the eye. So our hypothesis was that, similar to other groups, that if we can enhance the expression of the normal copy of PAX6, we maybe in part compensate for that insufficient gene dosage. And we're taking a drug repurposing approach, basically to take drugs that have been approved for other indications or clinically available, and to see if we can use them to enhance the expression of PAX6. So we basically started by doing a literature search for compounds and drugs that are known to have effects on PAX6 expression, and then used an in vitro system, basically primary human corneal epithelial cells, corneal epithelial cell lines, mesenchymal cells. So combination of different cells that we tested these compounds. But most of the data really was first focused on the epithelial cells. And these are the compounds, some of the more promising agents that we found that were able to increase the expression of PAX6 in culture: metformin, rosiglitazone, simvastatin. But MEK inhibitors or MEK inhibitors were the ones that we focused on more, given that they seem to consistently increase the expression of PAX6 at least by twofold. And some other drugs that actually reduced PAX6 were lithium and valproic acid. Again, maybe these drugs are promoting more of a proliferative phenotype. And again, this just shows that, this is from our paper that we published, showing that both at the mRNA and protein level, we can increase the expression, and we can get it with multiple agents. So MEK, or MAP kinase, as some of the scientists know, is what phosphorylates or activates ERK, which is part of a proliferative signal for cells typically. And previously, it's been shown by others that it negatively regulates PAX6. So we think it probably that MEK inhibitors are, at least in part by inhibiting ERK and proliferation, they're promoting more differentiation and enhancing PAX6 expression. And there are already several drugs that are approved or in clinical trials, and really the initial indication for most of these were for melanoma, but now they're being investigated in for other indications as well. So what we did in our animal model was we used this model, the small I new PAX6 mutant mice, which Jim kindly sent to us, Jim Lauderdale, with support from Vision for Tomorrow. And basically, we gave them either topical or oral MEK inhibitor, starting from day 5 after they're born, all the way through day 30. And basically, we looked at some of the main outcomes that we looked at were corneal clarity, thickness of the cornea, and thickness of the corneal epithelium. And basically, as you can see, this is the topical treatment group. This is the oral treatment group, wild type control, and the vehicle controls. We see thinner corneal stroma, thicker corneal epithelium, much closer to the wild type phenotype. And these effects were persistent at day 60 and day 90. And again, to correlate with that was the gene expression, where we again saw more PAX6 expression and more keratin 12 expression, a marker of corneal epithelium in the mice that were treated with the MEK inhibitor. And this just shows, again, the vehicle-treated group had much more proliferations, Ki67 staining, compared to the MEK inhibitor or the wild-type control. Similar findings were also found on ERG, where again we saw higher amplitudes in the MEK inhibitor treated group compared to control. Of course, still not as much as the wild-type controls. Also the same thing on visual function using optokinetic response, where there's definitely improved visual responses in the mice that were treated versus control. And also this sort of unpublished data from Sharong, who really we collaborated with on these studies and helped us a lot in getting these studies done with the visual function. And she has also found lower intraocular pressure in the mice that were treated with the MEK inhibitor during the first 30 days of life. Just a quick word: MEK inhibitors are actually interesting when they're given systemically. In some patients, it can cause this unusual adverse effect where they develop fluid in under the retina due to dysfunction in the RPE. It seems to be dose related, and when they reduce or discontinue the drug, it seems to go away, and no one really knows what's. So in summary, our studies seem to support this drug repurposing approach to try to enhance the expression of PAX6 in the setting of aniridia. Particularly if this could be done early in life, it may have the potential of slowing the progression of the disease. But of course, much more work is needed before this could get to the clinic to establish its safety. And also, as some of the other speakers probably will present or may have already presented, there are several other or many other candidates potentially that may be even safer than MEK inhibitors. Although at the doses that we're using, which were very low dose, we never saw any evidence of toxicity. Anyway, and this is the paper that was published late last year on some of the studies that I presented. With that, I want to thank everyone, thank all the lab members, the funding agencies, and collaborators, and thank the organizer again.